Hello again. Let's get into the eukaryotic cell. Let's start talking about some of the compartments that are the organelle systems found within the eukaryotic cell, and one of the major ones is called the endomembranous system. Now, the endomembranous system is a collection of interconnected membranes, and one of the key portions of it is the nucleus of the cell. The nucleus of the cell is one of the largest organelles found inside the cell. It has a double membrane surrounding it. It's separated, that double membrane separates the interior of the nucleus, what's called the nucleoplasm, from the surrounding material, the cytoplasm of the cell. The double membrane surrounding the nucleus is called the nuclear envelope. Now, we think of it as the control center of the cell, the nucleus. And that's an okay way to think of it. That's fine with me. But I think that you want to make sure that you make the connection later as we talk about the processes of genetics and protein production and what's referred to as transcription and translation, that you realize that the nucleus is controlling the cell through its ability to regulate the production of proteins. And it's the proteins that actually are the day-to-day controlling mechanisms used in the cytoplasm of the cell. So think about that as we get to that portion of material. So the DNA within the cell has information. That information is used to make proteins. Those proteins regulate cell function and structure. So here we have the nucleus, a double membrane structure, and you can see that the double membrane actually has a gap between it. You can also see that the membrane, the envelope that surrounds the nucleus, is penetrated by passageways called nuclear pore complexes. Associated with the membrane that makes up the nuclear envelope are the membranes that make up the endoplasmic reticulum, another component of the endomembranous system. Notice in this illustration it shows that the endoplasmic reticulum might have ribosomes attached to it, and that's why it's given the name rough endoplasmic reticulum, or rough ER. We're going to find that the cell has a number of these folded membranes, all right? Notice how they're interconnected with the membranes of the nuclear envelope. Inside the nucleus, what do I have? I have the DNA material in this unwound form. It's called chromatin. There's also a region within the nucleus called the nucleolus. That's a concentration of DNA and proteins, and the simple way to think about it is that it's the site within the nucleus where the building blocks of ribosomes are constructed, are first made. So notice, I'm not saying that ribosomes are produced there, but the building blocks of ribosomes are produced there. And as I mentioned, there's pores that allow materials to pass from the surrounding cytoplasm into the nucleoplasm, and vice versa, those nuclear pore complexes. All right, so many organelles are related. They're interconnected in this endomembranous system, and the nucleus is, in a sense, the central component of it. The endomembranous system is a collection of membranous organelles. They are focused on the manufacturing and distribution of materials inside the cell. And as we talked about before with membranes, these endomembranous system components subdivide the cell into compartments. You want to think of each compartment as having its own specialized aqueous solution inside of it. Remember, that's the whole idea of having these membranes, is to create barriers between distinct aqueous solutions on the two sides. It's essential for proper cell function. The endoplasmic reticulum is one of the key parts of this endomembranous system. What happens is that this rough ER, remember I mentioned the rough ER was called that because of the ribosomes attached to a surface, is primarily associated with adding to the membrane of other organelles, including the cell membrane, and in the production and packaging, initial packaging of proteins produced by the cell that are going to become part of the cell membrane or be released to the outside of the cell, or possibly those proteins are going to be used inside of other organelles. So, once again, ER, membranous system. Rough ER, ribosomes attached. Rough ER is focused on adding to the membrane material of other organelles, primarily by creating these vesicles, little bags of membrane that float off inside the cytoplasm of the cell and then fuse with other organelles. We're also going to see that this rough ER, because of the ribosomes associated with it, is the key site for the production of proteins used within other organelles that are part of the cell membrane, or proteins that are going to be released to the outside of the cell. All right, so the smooth ER, another portion of this endoplasmic reticulum, is smooth because it lacks the attachment points for ribosomes. So it doesn't have ribosomes studding its surface. The presence of those ribosomes is what makes the rough ER rough. If I have ER without ribosomes attached, I have smooth ER. Smooth ER is focused on carrying out the enzymatic activities to manufacture more lipids for the cells, in particular phospholipids, but also steroids. So a number of fatty acids are being produced by enzyme systems found associated with the smooth ER. Phospholipids are constructed out of these building blocks. Steroids are produced. Glycolipids are being made. A number of different lipid components are being produced through the actions of enzymes associated with the membrane of the smooth ER. In some cells, especially cells like, for example, your liver cells, the smooth ER also might be specialized for carrying out the detoxification, the breakdown of certain waste materials or toxic materials that might have been brought in from the outside into the cell. They also might be responsible for the degradation or breakdown of certain kinds of drugs or chemicals that are taken into the body. So there we have a representation. Notice where we are inside the cell. We have the situation where here's the plasma membrane, and notice what we're looking at. We're focused on the region along the surface of the nucleus. Nuclear envelope, right? Interconnected with the ER, both smooth and rough. Key difference between the smooth and the rough? Presence of the ribosomes. Notice the ribosomes that stud the surface of this rough ER. And the lack of ribosomes on the other side of the illustration where the smooth ER is. All right, so that's just one of the visible differences. We also mentioned that they functionally have differences as well. Now, associated with the ER, we're going to have another membranous structure called the Golgi Apparatus, or the Golgi body. Now, the Golgi Apparatus has as one of its key jobs taking proteins that were made by the rough ER, the ribosome along with the rough ER, and then modifying them, adding sugars to them, snipping off small portions, helping them finish to fold into their correct shape. And inside the Golgi also, proteins are sorted so that particular proteins can be sent to specific locations. Now, it sounds amazing, and the process itself is very interesting. In one of our courses, Cell Biology, we have the opportunity to talk about all of the molecular mechanisms that control how the Golgi sorts the proteins and sends them in vesicles and little bags of membrane to other organelles or to the cell membrane, where they're going to have their final job to carry out. This sorting process is fascinating. We won't have an opportunity to talk about it in much detail, but you want to think of that Golgi as being like a part of a company that receives materials, packages them, sorts them, packages them, and then sends them off to their destination. It's sort of like the mailroom of the cell. All right, so the Golgi apparatus. So notice where it's getting its materials from the ER, processing it, preparing it for sending to other locations. So notice the representation of the Golgi. Sometimes people refer to it as looking like a stack of plates. And you can see that it's an irregularly shaped multiple set of layers of membrane. As a matter of fact, each one of those little subcompartments of the Golgi, so if I was just talking about this one pancake-shaped, flattened double membrane structure, that's called a cisternae. And that word comes from the Latin word, which means a chamber. All right, and so we have these multiple layers of material, chambers of membrane. And notice how there's vesicles that are moving from the ER and joining with the Golgi, and moving from the Golgi to go to other locations, possibly to the surface of the cell, possibly to become part of another organelle. They might even be going back to fuse with the ER. So these vesicles, part of the endomembranous system, are very important for transporting materials from one location to another within the cell. Very specialized process regulated by large numbers of proteins and other molecules, as you might expect, and unfortunately not a topic we can really get into any of the details of right now. All right, but in general, what am I talking about? The Golgi or Golgi apparatus is a organelle, a structure composed of stack-like membrane containers called cisternae, and the key function of this Golgi is to receive materials from the ER, modify them, sort them, package them, ship them off to other locations within the cell, or ultimately maybe to the outside of the environment. All right, so what might be one of these places that one of those vesicles goes to? Might be for the formation of another endomembranous structure called the lysosome. Sometimes you'll see people just simply call a lysosome a digestive organ of the cell. Well, maybe not the best term to use for it, but here's the way we could think of it. It is a large vesicle, a large membrane-bound bag that contains within it large numbers of different types of digestive, breakdown enzymes. Things like hydrolases are found in great abundance in lysosomes. Now, the lysosomes are formed by vesicles that butt off of the Golgi and then fuse together to create this structure, this bag-like structure that has digestive enzymes. In a little bit, we'll see how those lysosome enzymes are used to digest materials, but right now we're just saying that this lysosome is a membrane bag, a large vesicle. It's a structure composed of a number of small vesicles fused together, containing high numbers of enzymes, especially enzymes that could be classified as hydrolases. All right. So now, what kind of enzymes do we find in this lysosomal mix? Well, they're going to be primarily those that are going to be digesting the food molecules, the external materials brought into the cell from the outside. They could also be enzymes that could be used to break down or destroy bacteria. If I'm a white blood cell and I'm consuming bacteria as part of an immune response of a human, then as I digest or I take inside that bacterial cell, one of the key things that's going to happen is lysosomes are going to pour their enzymes over that bacterial cell, kill it, break it down, destroy the bacterial cell. We find that lysosomes are also very important in recycling materials within the cell. They're not the only recycler, but they're one of the key ones. As a matter of fact, lysosomes can even wrap themselves around portions of other older organelles and break them apart. Reuse the pieces, but digest and recycle some of the cell's own materials. Now, one of the key things is that we find that lysosomes are important for the normal development of organisms because as an organism develops, it's producing large numbers of different kinds of molecules, many of which are produced at high levels at one time and need to be broken down so that then the cells can move on to carry out some new activity. Here's a basic diagram of this idea of materials being taken in from the outside and then processed, broken down. Notice, by the way, who the players are in this. Here's my rough ER. There's one of its transport vesicles that it sends to the Golgi. The Golgi apparatus processes the material and sends it off in the form of a vesicle. The vesicles enlarge, fuse together to become lysosomes. So let's follow what happens when one of these lysosomes interacts with a vesicle that was produced at the surface of the cell. By the way, this is a representation of the process called endocytosis. There's my food particle. You can see that it's being engulfed, taken into a vesicle-like structure formed by the plasma membrane. It's typically called a food vacuole. The word vacuole and vesicle really aren't distinctive names. You could think of them as being smaller versus larger. Vacuole being the larger sized chamber, membrane bag, vesicle a smaller membrane-like bag. So look what's happened. Food taken in from the outside is captured within a food vacuole. The food vacuole fuses with the lysosome. The lysosomal enzymes mix with that food molecule and break it down into smaller components. Those smaller components then can leave the lysosome, enter into the cell, and be utilized by the cell. If it were a organelle being recycled, you can see that represented also in this illustration. If this were a white blood cell killing bacteria, then the thing marked food would be something marked bacterial cell. So we're looking at a process where these endomembranous components, ER, right, vesicles, Golgi, lysosome, interact with vesicles that are bringing materials in from the outside to process and digest those resources. Now, interestingly enough, if I have a malfunction with my lysosomes, then that can lead to devastating disease process. Think about what we said about the embryo developing. The embryo is going to be very active, dividing cells, carrying out all kinds of specialized functions. As cells mature and specialize, they have to shift their activity. Since they're so very metabolically active, that means that they're processing stuff and needing to make materials and recycle them very rapidly. Well, what happens if my lysosomes aren't functioning properly? What if I have some kind of problem with one of the enzyme systems of my lysosome? Well, that's exactly the case that occurs in cells that lack an enzyme for breaking down a fatty acid that's produced by the cells. Because the cell can't break down the fatty acids build up to very high levels inside the cells. Eventually, the high levels of fatty acids that are produced cause the cell to have disrupted function, and eventually the cell dies. Now, one specific example of that is the disease called Tay-Sachs disease. It's an inherited, a congenital disease, where an infant born with the abnormality of one of these key lysosomal enzymes can't recycle the fatty acids, in particular in its brain cells, its neural tissue, and as a result, over a relatively short and unfortunately painful period of time, 6 months, 12 months, 18 months, the young individual, the infant, dies as a result of the buildup of these fatty acids. Unfortunately, there's nothing we can do about that. There is no treatment for the disease. So, this little organelle, the lysosome, and one of its many enzymes malfunctioning can lead to a devastating disease for an individual. Well, there's also larger membrane structures, especially found in plant cells, called vacuoles, and many plant cells have a very large central vacuole. In many cells, it's just for storing water, but in others, it might be used for storing all different kinds of carbohydrates. Some large vacuoles even function like the lysosomes of the animal cells. So another large membranous structure, a membranous structure we find in plant cells rather than in the animal cells. Interesting that there is a protist, the protista, have a specialized kind of membrane structure that's found in just some. This is a representation of a Paramecium, and inside of it you can see this elaborate membrane structure. Matter of fact, there's two of them noted. This is a membranous structure called a contractile vacuole. So it is a membranous bag, and it collects excess water inside of the cell. These are cells that live in aqueous environments, and they're constantly imbibing water. Water is flowing into the cells by osmosis, by diffusion. And so the cells typically have to try to get rid of this excess water. They do so with this specialized membranous vacuole called the contractile vacuole. As its name implies, once it fills with fluid, it then contracts and squirts that excess water to the outside environment. All right, so notice it's a type of pump, and it's a pump to get rid of excess water. Let's look at this endomembranous system in an overview from what we've just talked about. We started with the nucleus, double membrane making up its envelope. It's connected with the ER, rough and smooth ER. Production of materials, processing of materials takes place in the ER. Those materials are then sent to the Golgi. Golgi processes them further, sorts them, packages them, sends them off to other locations. Where? Outside of the cell, to become part of a lysosome, possibly to go back and rejoin the ER. So you can see that these membranes are all functionally interconnected, even though they may not be physically touching each other. They're all interconnected in terms of the roles they have within the cell. All right, so endomembranous system. key set of organelles, key functions in the cell. Beautiful example of compartmentalization, where I've taken the cell and put it into compartments, small, organized, specialized chambers that have specific specialized functions. Notice, however, how they interrelate with each other. We're going to look at some other organelles within the cell, but you can see that this endomembranous system is looking at a key functional component of eukaryotic cells in general. Thanks very much. Look forward to talking to you next time.